I'm Guy Maddern and I'm chairing today's session. We have two presenters as usual. I'd like to start by acknowledging the land on which we are positioned, the Kaurna peoples, and acknowledge elders past, present and emerging as part of our acknowledgement of their pivotal place in our society. This meeting today is focusing on CALHN and breast cancer. And the two presenters are based predominantly down at the Queen Elizabeth, but of course interact up to the Royal Adelaide as well. So if I could begin with Associate Professor Wendy Ingman, who is going to talk to us about improving breast health across the life course. It's all yours, Wendy.
Thank you, Guy. So the incidence of breast cancer in Australia has been rising for the past 30 years. And this is across all age groups. For example, there's been a 50% increase in the rate of breast cancer in premenopausal women. And this is against a backdrop of reduced mortality, and we have more and more Australians living well beyond a breast cancer diagnosis. And so the key questions for my research group is how do we reduce the incidence of breast cancer? And also how do we minimise the side effects of treatment for those who are diagnosed with breast cancer? Now, I did my PhD in obstetrics and gynecology, and I've brought to breast cancer research really the understanding that we have breasts over the life course, and there's an enormous number of developmental changes that occur across that time period, which are affecting breast cancer risk and also diagnosis and treatment. And I'm going to be talking about two of our projects today. One of them is looking at the pubertal growth period and how that affects breast cancer risk. And then the second project will be talking about menstrual cycling and how that's affecting breast cancer diagnosis and treatment. So puberty is a critical stage for breast development. This is when the endocrine and local factors converge on the breast and promote the development of the epithelium, the stroma and the adipose tissue, which then are the key components of the breast during adult life. And if we can actually measure the different components of the breast in the adult through looking at the radiographic appearance of the breasts on a mammogram. And so what you can see down the bottom is an X-ray of a breast section, which shows some white areas and some darker areas. And when we do an image-guided biopsy of those areas, we find that they are composed of different proportions of epithelium, stroma, and adipose tissue. So low mammographic density is primarily adipose tissue, and high mammographic density is primarily stroma and epithelium. And these actually affect the risk of breast cancer over a woman's lifetime. Mammographic density is an independent risk factor for breast cancer. Women with extremely dense breasts, seen here on the right, will have a 4 to 6-fold increased risk of developing breast cancer compared to women with mostly fatty breasts, seen on the left. And this is when matched for age and BMI. And epidemiological studies have shown that there is an association between how breasts grow during puberty and the adult breast cancer risk and mammographic density. And this is an inverse association. So when girls have a higher pubertal weight or BMI or slightly more adipose tissue on their bodies as they're going through puberty and breast development, they actually have a reduced adult breast cancer risk and reduced adult mammographic density when adjusted for adult BMI and the timing of menarche. So this is an epidemiological association. It hasn't been shown to be causative. And so our research started to try and address that question. Is the amount of adipose tissue put down on a girl's body during puberty affecting her adult density and breast cancer risk? And we have begun looking at this in an animal model. So this is the ALMS model of pubertal adiposity. So these mice have a hyperphagia, so they overeat and they will gain weight compared to their wild type littermates, even if they're fed a healthy diet. And in our model, these mice are allowed to overeat or eat ad lib during their pubertal development, so between 3 and 7 weeks of age. And then they are adjusted so that they can have normal adult weight. So we have paired feeding for these mice. So we have control mice, which eat normal amount of food, the ALMS null mice, which are given the same amount of calories as the control mice. And then an additional control, which is the ALMS null mice, which are allowed to feed ad lib throughout the entire experiment, and they are obese as adults. And we can cross these mice with a mouse model of mammary cancer to look at their cancer progression. And what we find is that mice that have developed during pubertal adiposity have altered mammary gland density. So they have less fibroglandular tissue and more adipose tissue compared to controls, and reduced abundance of collagen around that epithelium as well, which is reminiscent of what we would call mammographic density when conducted a mammogram on humans. And when we look at their development of tumours in the PYMT mouse model of mammary cancer, we see that the pubertal adiposity mice will have reduced weight of the primary tumours and reduced tumour burden and reduced number of tumours. And they also have a delayed onset of tumorigenesis as well. So this is really suggesting that pubertal adiposity during that key mammary gland developmental period is actually affecting the adult mammary gland growth and tumour development. So we've also been looking at adult menstrual cycling and how that affects a breast cancer diagnosis. And the menstrual cycle is really underappreciated in premenopausal breast cancer. Clinicians don't record menstrual cycle stage when they are looking at their diagnosis or treatment of breast cancer, but there really are very significant fluctuations in estrogen and progesterone over the course of 28 days, which are particularly going to affect the function of the breast and the breast cancer if it is hormonal receptor positive. And this is relevant because there are new genomic assays which are emerging in the clinic which are helping to guide treatment decision making. And leading genomic tests are Oncotype DX and Prosigna that evaluate the expression of gene biomarkers and then formulate them into an algorithm which predicts the benefit of adding chemotherapy to endocrine therapy. And these are tests which are helping patients to avoid chemotherapy if it's been shown that they have low risk of recurrence and therefore can avoid those chemotherapy-associated toxicities. However, these genomic tests were developed predominantly in postmenopausal women. And so we have here a table showing the Oncotype DX development and clinical validation conducted predominantly in postmenopausal women, and premenopausal women didn't feature very strongly in its development. But when we look at the mouse mammary tumours in the PYMT model and compare two different stages of the ovarian cycle, so the estrous stage, which is reminiscent of the follicular phase in humans, versus the diestrous phase, which is reminiscent of the luteal phase, we see a lot of changes in gene expression of these key genes which are used in the Oncotype DX test. And when we calculated an experimental recurrence score, we found that the mammary tumours which were taken from mice at diestrus actually had significantly higher scores than mice that were taken at estrus. And when we looked at human paired samples, we found something similar. So we didn't know the menstrual cycle stage of these patients, but what we were able to do was a paired sample analysis. So typically diagnosis and a core biopsy is conducted about 2 weeks before surgery and excision of the tumour, which gives us 2 samples taken 2 weeks apart, which we can compare to see whether there is discordance in those recurrence scores and how age affects that, which we would expect that a postmenopausal woman who is not having menstrual cycling anymore will have better concordance between these two samples than a premenopausal woman who's still undergoing menstrual cycling and will have a difference in her hormone profile at that time. And so what we see on the left is the relationship between sample 1 recurrence score and sample 2, so the diagnostic core versus the surgical sample. And generally there was good concordance, but you can see that there are some outliers there which are further away from perfect concordance than others. And when we look at the difference in recurrence score as a factor of age, what we see is that there is better concordance in those two paired samples for older women and worse or discordance when we look at younger women. And when we break that down, what we see is that the genes which are driving this discordance in these younger women are genes associated with proliferation and HER2. So this was the first evidence that the menstrual cycle could indeed be affecting these genomic assays and potentially even the treatment decisions which might stem from their use. We've looked in the mouse model to evaluate whether it's menstrual cycling which is affecting the discordance in the paired samples in younger women. And further research is needed now. We're thinking of this really as a pilot study. It was 25 samples, and we're hoping to increase this research and look at this further. And this has been a huge team which has been involved in this work. We work collaboratively with an enormous number of people at the Queen Elizabeth Hospital. So we had to fill up the whole page with this enormous list, and we worked with some wonderful collaborators in Roseworthy and in the Adelaide Medical School as well. Thank you.
Thank you very much, Wendy. This is really interesting work, and I'm never quite sure where the epidemiology is going to match the science, or at least the practical science. But these Oncotype DX and Prosigna, they're expensive, aren't they? And I understand they're not yet available as a part of the Medicare schedule. Am I correct?
That's right. So Oncotype DX costs $5,000. Prosigna costs about $3,000. And there have been a number of applications for their inclusion in the for benefits, and they've been denied each time. And that's largely because the MSAC requirement is that the test is only going to be eligible for rebates if it's proven to be better than current standard of care, and they don't believe that that evidence is strong enough to show that what Australian doctors are doing right now in terms of deciding who gets chemotherapy is actually going to be greatly improved.
Yeah, no, it's, I mean, I guess it will take time, and of course my understanding is most of these tests have to be sent offshore to be done, which adds a lot to the cost.
Yes, so Oncotype DX has to go to America, and then it's not just the cost, it's also the turnaround time, so it will take 3 weeks to get the answer.
Right, right. Well, look, thank you very much, and we will continue on now. If I could introduce our next speaker, who's Betty Sallustio, who's going to talk to us on new therapeutic approaches to preventing heart damage during chemotherapy in breast cancer patients. Betty, the floor is yours, and try and share your screen.
Thank you. I will try and share it correctly. I'm sort of a last minute fill-in for Andrea Sevdocchio. So I thought I would talk about a research project that we've been conducting in collaboration with the Breast Cancer Research Unit, which is looking at new therapeutic approaches for preventing heart damage following cancer chemotherapy. And this project focuses on the anthracyclines, which are an important group of cytotoxic agents used for the treatment of breast cancer. It's actually part of a much bigger research program, which we're conducting with the Breast Cancer Research Unit, with the Cardiovascular Pathophysiology and Therapeutics Unit, and with the clinical echocardiographers, and it's being conducted here at the Basil Hetzel Research Institute. So chemotherapy-induced cardiovascular disease is an important concern in breast cancer survivors because many of the therapies that are used to treat breast cancer are cardiotoxic. Some can cause acute changes in heart function that occur during therapy, and these changes can result in chemotherapy cycle delays, reduction in the intensity of chemotherapy, or cessation of the cardiotoxic therapy, all of which can increase the risk of treatment failure and cancer recurrence. Some chemotherapy agents can cause progressive long-term myocardial damage, such as heart failure, which can then become a leading cause of morbidity and mortality in cancer survivors. So given the improved breast cancer cure rates and longer survival of patients, there is increased focus now on preventing the long-term adverse effects of cancer therapies. And reducing the risk of progressive cardiomyopathy and heart failure is particularly important in the growing cohort of young breast cancer survivors. Okay. All right, so why can't I go on to the next slide? Aha. So this, the anthracyclines, particularly doxorubicin and epirubicin, are widely used for the treatment of breast cancer. And although they're very effective, they present one of the biggest risk factors for cardiotoxicity. This slide shows the exponential dose-response relationship between cumulative anthracycline lifetime exposure and the risk for cardiomyopathy. And you can see in adult patients that receive high doses of anthracyclines, they have approximately 30-fold higher chance of developing heart damage compared to matched patients who do not receive anthracyclines. So clinical guidelines for the prevention of cardiac damage in adult cancer patients highlight anthracyclines, radiotherapy where the heart is in the field of treatment, and the use of trastuzumab as the 3 treatment modalities that confer the greatest risk of heart damage. And importantly, anthracyclines aren't toxic or cardiotoxic just at the high doses. They can contribute to cardiotoxicity even at lower doses, where, for example, when they're used in combination with radiotherapy, or when they're used in patients who already have risk factors for heart disease, who are older, or who already have compromised cardiac function, or when lower dose anthracycline treatment is used sequentially with trastuzumab therapy. So how do anthracyclines work? Anthracyclines inhibit topoisomerase II enzymes. These are enzymes important in DNA replication and transcription, and they work by causing transient double-strand breaks in the DNA, which allows uncoiling of the helix and relaxation so that replication and transcription can take place. The anthracyclines bind to the DNA and they form a complex, an anthracycline topoisomerase DNA complex that then prevents repair of that double-stranded break. So they cause DNA damage, they prevent cell replication and DNA transcription, and they cause apoptosis. And their clinical toxicity reflects inhibition of cell proliferation, for example, gastrointestinal toxicity and myelosuppression. And whilst myelosuppression is an important cause of cycle delay, it can be managed medically. And cardiotoxicity is the most important dose-limiting adverse effect of the anthracyclines. So adult cardiomyocytes are not regenerative, and so damage that causes myocyte death accumulates and will eventually overwhelm the heart's adaptive mechanisms. We know that DNA damage, mitochondrial damage, and apoptosis are involved. However, the mechanisms have not been fully elucidated. Now, the central role of apoptosis suggests that a naturally occurring cytokine called TNF-related apoptosis-inducing ligand, otherwise known as TRAIL, may play a key role in anthracycline cardiotoxicity. So TRAIL is a key activator of the extrinsic apoptotic pathway. So TRAIL is an agonist of death receptors that are expressed in humans on cell membranes, death receptor 4 and death receptor 5. These death receptors have a death domain, and following the binding of TRAIL, this activates caspases and the downstream pathways of apoptosis or programmed cell death. Human cells also contain decoy receptors. There are membrane-bound decoy receptors, 1 and 2, which can also bind TRAIL, but these decoy receptors don't have the death domain, so they can't induce apoptosis. And there are also soluble decoy receptors such as OPG, which can also bind to TRAIL, but can't induce apoptosis. So the decoy receptors compete for binding to TRAIL and thereby can decrease apoptotic signaling. So we have now put together quite a bit of in vitro evidence to show that TRAIL signaling is essential for the cardiotoxicity caused by doxorubicin to heart cells. So in using cell culture, we've shown that doxorubicin on its own doesn't cause cytotoxicity, but in the presence of TRAIL, there is significant loss of heart cell viability at low micromolar concentrations of doxorubicin. Heart cells seem to be particularly sensitive to the effects of doxorubicin and TRAIL, whereas TRAIL on its own causes no loss of viability. And in contrast, other cells, such as human skin fibroblasts, they are not susceptible to cytotoxicity of DOX and TRAIL. Now, we've also shown that doxorubicin can increase the expression of the death receptor 5 on the cell surface, and this points to a potential mechanism for the increased cell death caused by both doxorubicin and TRAIL. We've shown that it's possible to prevent doxorubicin and TRAIL cardiomyocyte toxicity in vitro, and we've done this by co-incubating with 2 decoy receptors, either the soluble OPG in red or the soluble DR5 receptor. So when doxorubicin and TRAIL are given together, they cause significant loss of heart cell viability compared to control cells or doxorubicin alone, but this can be reversed in the presence of the soluble OPG, which can mop up TRAIL, and also the soluble death receptor 5. And with our PhD student now, Michelle, we've gone on to show that TRAIL is essential for doxorubicin-induced cardiotoxicity in vivo. These studies use wild-type mice and TRAIL knockout mice that were administered either doxorubicin or saline every day for 21 saline every 5 days for 21 days. And then heart function was assessed by a blinded echocardiographer. And we measured left ventricular ejection fraction and fractional shortening. And these are the results. You see, there's a similar effect on both measures of heart function. In wild-type mice, doxorubicin, here shown in blue, doxorubicin significantly affected heart function, causing a significant decline. But the knockout mice, shown in red, were protected from the cardiotoxic effects of doxorubicin. In collaboration with a Chinese group who are providing us with soluble death receptor 5, we have now shown that it's possible to prevent acute doxorubicin-induced heart damage in vivo in wild-type mice. And this by injecting soluble death receptor 5 intravenously. And these are the results, again, for ejection fraction and fractional shortening. The mice that are administered doxorubicin, you can see there's a significant decline in heart function compared to controls. But the mice that are treated with soluble death receptor 5, as well as doxorubicin, heart function is preserved. And histological analysis of the hearts show that this preservation of heart function is because there is less cardiomyocyte death in the animals treated with the soluble DR5, and this has been measured by tunnel staining. So our working model for doxorubicin-induced cardiotoxicity is that doxorubicin upregulates the expression of death receptors in cardiomyocyte cell membranes, and then it is the increased trail signaling that causes apoptosis and cardiomyocyte cell death. So we now have the opportunity to develop completely novel cardioprotective agents that protect the heart by inhibiting trail signaling. And we may be able to do this using neutralizing antibodies to trail, or using soluble death receptors or soluble decoy receptors to mop up trail. And we hope that this may not only be useful as a cardioprotective strategy in oncology, but perhaps in heart disease in general. And finally, I'd like to thank our collaborators in New South Wales, Dr. Mary Kaburma, who provided the knockout mice, and the Chinese group who are providing the soluble death receptor 5, and our PhD student, Michelle Sens. Thank you very much.
Betty, thank you very much. Can I just ask, as a clinical issue, and I realise you're not necessarily talking to us about that, how do we detect the cardiac damage of these agents? Is this something that's picked up on ECG, on blood? How do we know that it's occurring?
At the moment, it's done with echocardiography. And that's, I believe, that at-risk patients would be screened during their breast cancer treatment and then at various periods following. But the progressive cardiomyopathy and heart failure is really hard to predict. You can have completely normal echocardiography during treatment and then develop heart disease years after.
Yeah.
So.
And, yeah, no, it's and how common is this condition? Not the breast cancer, obviously, the chemotherapy-induced cardiac things. Is this happening in 1% of patients or 20%? Do we know?
The data in adults aren't as good as in children. The young survivors of cancers, there's a lot more data on. I think it's about 1 in 10 breast cancer survivors have evidence of cardiac dysfunction in the first, I think it's the first 5 years after treatment.
Okay, so it's certainly not an insignificant problem.
Not necessarily frank heart failure, but echocardiographic evidence of cardiac dysfunction.
All right. Well, look, we have fully used our time, and you fully used your time. We have no other questions. So look, on that note, I will thank you, both of you, very much for giving really fascinating talks in a very important area. We will be back again next week with another Adelaide Biomed City mini review, and hopefully we will gain the attendance of those who are here today and others. So thank you very much, and we'll see you again next time.